Hi everyone, welcome back to my YouTube channel. Don't forget to like, share, and subscribe. Microbial genetics, we have our chromosomes. When we say chromosomes, it is one long double helical strand of DNA. The DNA is tightly wound up and associated with many DNA-binding proteins. These proteins are essential for carrying out. We have here two functions of DNA, the replication and the expression. The expressions, which can be divided into two parts, the transcription and translation. The DNA binding proteins include DNA polymerase, RNA polymerase, endonuclease, and ligase, gyrase, zinc fingers, helicase, leucine zippers, and histones in eukaryotes. When we say genome, all the genetics that a cell must have functions and reproduce as a cell of a particular species. The human genomes consist of 22 different autosomal chromosomes and have two different 6 chromosomes. Moreover, each human cell has a pair of each autosomes and a pair of 6 chromosomes. Therefore, human cells are said to have a diploid genome and the 4 bacteria have a haploid genomes. Genes, it is a sequence of DNA which codes for a functional product. The product is usually a protein of some kind like an enzyme or a receptors or a structural protein. Genotypes, this refers to the genes which an organism actually possesses. This is the potential of organisms. The phenotype, this refers to the genes which are actually expressed by the organisms. This is usually what the organisms looks like. Allies are the different varieties of genes which exist in the population of the organisms. How DNA functions. Remember that DNA is double-stranded. The two strands are complementary and the strands are hybridized to each other because of the base pairing. DNA replication will need DNA polymerase, the RNA polymerase, helicase, and gyrase, and ligase. The replication process ensures that when DNA is replicated, the two exact copies will be made. If a mistake is made, it is called a mutation. A mutation is an inherited changes in a nucleotide-based sequence. We have heard the protein synthesis. First, we have transcription. In transcription, we have the DNA copied into mRNA. RNA polymerase binds to the promoter and synthesis mRNA from the strand of DNA. Many copies of mRNA are made. RNA polymerase drops off when it hits the termination region. The second one is the translation. mRNA is read and used as a template to make a protein. This 5' end of the mRNA binds to the small subunits of the ribosomes, and then the large subunits bind to that. The AGU is always the first codon read in the translation. There are 61 different tRNA. This refers in their anticodon sequence, which can hybridize with the 61 codon specified amino acid. The mRNA binds to the ribosomes so that the first codon binds to the P site and the second codon is in the A site. When a nonsense of stop codon enters the A site, the polypeptide is cut off of the last tRNA and both are released. In bacteria, one ribosomes begin to move down and mRNA or another ribosomes can bind to the promoter. And as this one moves downstream, still another ribosomes can bind. So we have the two types of mutation at the DNA level. The base substitution point mutation, the second one is frame shift and insertion or deletion. When we say gene regulation, the lactose operon is a group of structural genes which are controlled by the one operator, or the lactose operon is repressed and by glucose and induced by lactose. So those are the microbial genetics is that I discussed to you. That's all. Thank you.